making a quick recording to go over the syllabus real quick for anybody that needs to look back at it or anything. So for the Molecular Diagnostics class, summer 2025, this was a 2-hour class. My office hours are 9 a.m. to 12:30 on Mondays and 1 p.m. to 5 o'clock after lunch. And then Tuesdays, 1 o'clock to 3:30 in the afternoon. But I'm available other days and times if you need me. So just feel free to reach out to me and we can meet up if we need to, or just do a Teams call. Alright, so student learning objectives. After this course, you'll be able to describe the basic structure of DNA and RNA, describe the basic process of DNA replication, perform DNA extraction on a clinical specimen, which we'll be doing this coming Tuesday. Be able to demonstrate an understanding of basic molecular diagnostic techniques, demonstrate understanding of electrophoresis and the separation of DNA fragments, and also be able to apply molecular diagnostic techniques to the identification and diagnosis of diseases. In fact, the last lab that we do will be really fun. We'll pretty much be analyzing DNA found at a so-called crime scene, and of our the subjects that are suspects for the crime, we're going to see whose DNA matches the crime scene's DNA, which doesn't necessarily mean that they did it, but that at least means that they were present at the crime scene. So that's what we'll be doing toward the end of the class. When it comes to the grade breakdown, we've got lecture grades. That'll be 4 exams. There's going to be an exam each week since this is just a 5-week course. Those exams will count 20% each of the lecture grade, and then the quizzes will be 20% total altogether. And then when it comes to the lab grade breakdown, 50% is just lab participation. So if you turn in your worksheets at the end of the labs, you'll get 100 for that. And then the other part is 50% homework average, which will be media lab homework. And then for the final grade, it's a 75-25 split for the lecture versus lab grade. Got to make a C in both components to pass the course. When it comes to required instructional materials, there's not a book that's required for this class. When it comes to in the optional instructional materials, that Leela Buckingham book, that's kind of what we're going based off of, but there's way more than what y'all have to know for basic molecular techniques. So pretty much Mr. Barrett in the past had put together just little lecture Word documents of what you really need to know. It's just basic stuff. But if you are super interested in molecular diagnostics, I do have a couple of these Leela Buckingham books that you could borrow if you want to. And next we'll go down to course structure and overview. So it'll be per week. There'll be 2 lectures that are gone over. So we'll look at that more in just a minute. Per lecture, there will be a quiz, and these quizzes will be completely based on the study guide that y'all will fill out. These study guides are not for a grade, but if you fill out the study guide, these are exact question answers, or at least directly related to what's being asked in the study guide. So if you fill out those study guides, you'll do really well on the quizzes. When it comes to the quizzes, it's like how I've had it in the past, where you can use anything that you want. It's open source, and you can take the quiz multiple times, but only the first attempt will be what's recorded for the grade. So I do highly suggest having that study guide right next to you for those quizzes. When it comes to the homework grades, those will be the Media Lab assignments. Each week there will be a Media Lab, and there will also be a quiz over that Media Lab. So make sure you do the quiz over the Media Lab. I mean, it's going to be stuff that's pulled directly from questions inside the Media Lab, so you can have Media Lab open while you're doing that quiz if you want. So yeah, there will be spots to upload your certificate of completion for those Media Labs. So those will count for the homework grades. So make sure to do those. And so per lecture, there's going to be a quiz, and there's always going to be a Media Lab quiz each week. So for example, for this coming Tuesday, what's going to be due before class is a Lecture 1 quiz, a Lecture 2 quiz, a Media Lab certificate of completion, and a Media Lab quiz. So 4 different things. And so then let's move on to exams. There'll be an exam each week since it's such a fast-paced course. These will be directly taken from the quizzes, and like the lecture quizzes and the Media Lab quizzes. So make sure you study both types of quizzes. So this coming Tuesday, questions will be pulled from the Lecture 1 quiz, Lecture 2 quiz, and the Media Lab quiz. And then there'll be lab worksheets to do in lab, so that's real simple. When it comes to communications, of course, y'all can Teams me or text or call me, whatever you want, or email me. Let's see. Just when it comes to attendance, please try to be at every single meeting that we have. I mean, we've only got 5. I mean, so it's really important to be there. I mean, if there's some extenuating circumstance, I understand, we'll figure it out. But really try to make it each Tuesday, because it's really important to be there. So more than one unexcused absence will result in the student being dropped from the course. 2 unexcused late class attendances equals 1 unexcused absence. I don't think anybody's going to be late in this class. But when it comes to makeups, I will accept late work for this course, unlike the spring semester, because it is such a fast-paced course, and there's not a whole bunch of grades to make up that final grade. So every grade counts, quite a bit. So if you'll see in the course schedule that things are due, like normally I would have things due Sunday at 11:59 p.m. For this course, since it's so fast, I'm actually giving a little bit more time. You just have to do this week's material by the following Tuesday before class, so like before 9:30 a.m. Tuesday. So that's a little bit of extra time. But if there's some weird reason that you don't get your homework in by Tuesday before 9:30 a.m., I will take it late up till Thursday at noon at a 10% penalty for being late. So just letting you know that's out there in case any of y'all end up in that situation, but do try to, have those on time, because each grade is going to count quite a bit. So yeah, that's pretty much it for the syllabus. Now let's look at the course schedule real fast. So the breakdown, we've got, only 5 weeks here. So what we did for this first week was lectures 1 and 2 on Blackboard or under the week 1 tab. The quizzes for each of those lectures, which are like little, basically like typed up documents that Mr. Barrett had made a few years ago. There are quizzes for each of those. There's a study guide to fill out for each of those, which will help you with these quizzes, but is not due for a grade. And then there's a spot to put in the MLAB homework certificate of completion. And so, yeah, you'll have 4 things due by Tuesday at 9:30 a.m. And then for lab, we just did that quick little, virtual lab, and kind of just went over some things that, when it comes to, like practical applications in the lab that y'all would probably mess with, things like the BioFire, or the Luminex Aries instrument. Also, Verigene's a really nice multiplex array test made by Luminex. So we had that little worksheet that we did in class, and then I'm going to be uploading later a video of me doing the lab that we're going to be doing this coming Tuesday, because we do have to share resources on it. Like, they only give us one protease reagent, like, within this DNA isolation kit. So it gets a little bit confusing, like, sharing and, after so-and-so is done, can I have 20 microliters of protease, and then I need this or that. It gets a little confusing, since we're going to have to